Hi, this is Jeff Hoffman, and welcome to the nutrition section of biochemistry. Vitamins are needed for certain enzymes to work, and for the step 1, you should know which enzymes require which vitamins, the main source of each vitamin, and what clinical symptoms are caused by excess or deficiency of each vitamin. You don't need to know their molecular structure. We'll break the vitamins down into two categories, fat-soluble and water-soluble, because each class is absorbed and stored in a different way. The fat-soluble vitamins are A, D, E, and K, and they are absorbed in the ileum of the small intestine, since that's where fat is absorbed, and they have to be absorbed with fat. Since fat requires pancreatic enzymes to be absorbed, patients who have a pancreatic enzyme deficiency can't absorb fat-soluble vitamins and can become deficient. If fat isn't absorbed, it will be passed out in the stool, which is called steatorrhea, and the vitamins will go with it. Can you think of an autosomal recessive disorder in which this happens? The main example you should think of is cystic fibrosis, in which thick mucus plugs prevent pancreatic enzymes from getting to where they're needed, and fat-soluble vitamin absorption is prevented. In healthy patients, fat-soluble vitamins enter the lymphatic system after being absorbed, and eventually end up in the blood. Now, since they're not water-soluble, how do they travel in the blood? They can only travel in the blood by associating with carrier proteins such as albumin. Ultimately, they will accumulate in fat, so it takes a long time for them to be cleared from the body. This is why it's much harder to become deficient in fat-soluble than water-soluble vitamins, and also why it's easier to accumulate toxic quantities of them. The water-soluble vitamins include all of the B vitamins and vitamin C. Unlike the fat-soluble vitamins, the water-soluble ones are absorbed directly into the blood. They're also excreted very easily in the urine, except for B12 and folate, which are stored in the liver. This is why it's relatively easy to develop deficiencies of all of the water-soluble vitamins except for B12 and folate. We'll go through the symptoms of deficiencies for each vitamin, but in general, a deficiency in most B vitamins can cause dermatitis, glossitis, and diarrhea. Glossitis means a red beefy tongue, which you can see in this image. Vitamin A, also called retinol, is a fat-soluble vitamin found in liver and leafy green vegetables. After being absorbed from the intestine, it's transported to the liver within chylomicrons, and to the rest of the body by attaching to prealbumin and retinol binding protein, or RBP. It functions as an antioxidant, and it's a key part of a molecule called retinal, which is produced in the retina of the eye, and is the primary molecule used to convert light into a signal. This is why vitamin A deficiency can cause night blindness, also known as nyctalopia. It's also essential for the differentiation of epithelial cells into other tissue types, such as pancreatic cells, mucus-secreting cells, and sebaceous glands. This last one is why it can be used as part of a formula, such as Accutane or Retin-A, to treat acne. This is also why its deficiency can cause dry skin. It can regulate cell differentiation because it binds to and activates a protein called retinoic acid receptor, or RAR. When bound to vitamin A, RAR can form a heterodimer with RXR, or the retinoid X receptor, and then this heterodimer can bind to specific DNA sequences to activate transcription of genes, which cause the cell to differentiate into a different cell type, like a pancreatic cell. You might be interested to know that the thyroid hormone receptor and the vitamin D3 receptor also regulate transcription by becoming activated by thyroid hormone and vitamin D, respectively, and then forming a heterodimer with RXR. Vitamin A can also be used to treat some diseases. Extremely large megadoses of vitamin A, about 400,000 IU per day for 2 days, reduces mortality in children infected with the measles. It can also be used to treat acute myelocytic leukemia type M3, since again it will bind to the retinoic acid receptor, heterodimerize with RXR, and basically force transcription of genes that cause the promyeloid cells to differentiate, thus treating the cancer. Pretty cool, huh? Unfortunately, you really have to watch out for vitamin A during pregnancy, since its role in expression of differentiation genes, such as the Hox genes that you'll learn about in the reproductive chapter, make it a teratogen. This is why you need a negative pregnancy test and a discussion about contraception before prescribing any vitamin A-based medications, such as for acne. In addition to being teratogenic, acute toxicity with vitamin A can cause nausea, vertigo, and blurry vision, while chronic vitamin A excess can also cause arthralgias, fatigue, headaches, and a sore throat, although in general it's pretty well tolerated. Alright, time for our first flash quiz of this section. So this should be a pretty easy one hopefully, but you really do have to know these. So what are the fat-soluble vitamins? So these are A, D, E, and K. And if you don't know those yet, try to memorize them soon. Vitamin B1, or thiamine, is a water-soluble vitamin that's found in grains, meat, and legumes. B1 functions as a cofactor for several enzymes in the form of thiamine pyrophosphate, or TPP. This is formed by ATP donating two of its phosphates to thiamine, which are these phosphates here. And by the way, you should not confuse thiamine with thymine, which is the base of a nucleotide. The enzymes that require TPP as a cofactor are pyruvate dehydrogenase, alpha-ketoglutarate dehydrogenase, transketolase, and branched-chain amino acid dehydrogenase. These are all covered in more depth later in the chapter, so I won't go over their specific functions now. But do you remember what dehydrogenase enzymes do? They catalyze oxidation-reduction reactions. The most common cause of B1 deficiency in the U.S. is alcoholism, since alcohol prevents B1 uptake in the small intestine. When B1 is deficient, these enzymes can't function, which is especially problematic because glycolysis and the TCA cycle partially shut down. This reduces ATP production and causes an increase in lactic acid. So do you think it would be a good idea to give these patients extra glucose? This would actually hurt them because it can cause Wernicke's encephalopathy, which I'll go over in a minute. To prevent this, you should give extra B1 before giving them glucose. So not being able to produce enough ATP is going to have the biggest effect on tissues that need lots of ATP, which include the brain and the heart. This is why B1 deficiency causes Wernicke-Korsakoff syndrome. The Wernicke part of this is an encephalopathy that includes ophthalmoplegia, nystagmus, truncal ataxia, and confusion. If this is not treated, the Korsakoff part will kick in and includes less reversible symptoms such as impaired short-term memory, confabulation, and personality changes. These symptoms are due to damage caused by ATP deficiency in the medial dorsal nucleus of the thalamus and the mammillary bodies. Another consequence of B1 deficiency is called dry beriberi. This is a motor and sensory neuropathy that manifests as polyneuritis and symmetric muscle wasting. And the muscle wasting is really apparent in this picture of a man with dry beriberi, again caused by B1 deficiency. A third consequence is wet beriberi, which is both a neuropathy and a high-output cardiac failure, which results in dilated cardiomyopathy, and you can see that in this picture of an enlarged heart with thin ventricular walls. Patients experience peripheral vasodilation, biventricular failure, and edema. I also want to mention that patients with HIV are deficient in B1 much more commonly than the general population, which is presumed to be the case because of the cachexia and catabolic characteristics associated with AIDS. Therefore, patients with HIV are also at risk for Wernicke-Korsakoff syndrome. Vitamin B2, or riboflavin, is often found in dairy products and is a cofactor in many oxidation and reduction reactions. Most importantly, it's a precursor to both the FAD and FMN molecules, and in this form it can add or remove 2 electrons at a time from other molecules in a redox reaction. This is how it's involved in fatty acid oxidation, amino acid oxidation, and aerobic metabolism. It's also a cofactor for erythrocyte glutathione reductase and for the conversion of tryptophan to niacin, which is vitamin B3. Do you remember what glutathione reductase does? It reduces GSSG, which is the oxidized form of glutathione, to the reduced sulfhydryl form, which is an important antioxidant. Since erythrocytes are very sensitive to oxidative damage, patients who can't reduce glutathione due to vitamin B2 deficiency can get anemia. Deficiency in vitamin B2 can also cause the 2 C's, which are chelosis and corneal vascularization. Chelosis is inflammation of the lips, with scaling and fissures at the corners of the mouth, as you can see here. Vitamin B3, or niacin, is found in liver, milk, and grains, and with the help of vitamin B6, it can also be synthesized from tryptophan. Similar to B2, it's involved in redox reactions, since it's a constituent of NAD and NADP. NADH provides the energy to form 3 ATP molecules in the electron transport chain, which you can remember because niacin is vitamin B3. Now, niacin can also be synthesized from tryptophan, which requires the cofactor vitamin B6. So niacin deficiency can be caused by a Hartnup disease, in which there is decreased tryptophan absorption. Since niacin can be derived from tryptophan, less tryptophan means less niacin if you're not getting enough from your diet. Deficiency can also be caused by malignant carcinoid syndrome, in which tryptophan is being used up to make serotonin. Lastly, the drug isoniazid, or INH, prevents the absorption of B6, and therefore prevents the synthesis of niacin from tryptophan. Moderate niacin deficiency can result in glossitis, which is a thickened, inflamed tongue, as you can see here. More severe deficiency can cause the 3 Ds of pellagra, which are diarrhea, dermatitis, and dementia. Extremely severe deficiency can cause the 4th D, or death. So how do you think you can treat pellagra? All you have to do is administer niacin and tryptophan. Excess of niacin can cause facial flushing. You'll probably never see this in someone who just ate too much liver, but it can happen in patients who are prescribed large doses of niacin. Niacin can be given pharmacologically to treat hyperlipidemia since it lowers LDL and raises HDL. While it sounds like this would be pretty useful, it's actually rarely used since you have to give so much niacin that you get some unpleasant side effects, which include pruritus, hives, nausea, and vomiting, in addition to the facial flushing I already mentioned. These side effects can be reduced with aspirin. Alright, time for another flash quiz. So what are two symptoms of vitamin B2 deficiency? Remember, the two C's of B2 deficiency are chelosis, which is scaling around the lips, and corneal vascularization. Vitamin B5, or pantothenate, is used to make coenzyme A, which is a cofactor for a lot of the metabolic reactions that we'll cover in the next section. At the end of glycolysis, pyruvate dehydrogenase can make acetyl coenzyme A from pyruvate and coenzyme A. As you can see, this is necessary for the TCA cycle, fatty acid synthesis, and cholesterol and ketone body synthesis. B5 deficiency is pretty rare, but it can cause dermatitis, enteritis, alopecia, and adrenal insufficiency. Vitamin B6, or pyridoxine, is found in meat, egg yolks, and wheat. Its active form is pyridoxal phosphate, which is used in transamination reactions, which is when you move an amine group from one molecule to another, such as the liver enzymes ALT and AST. ALT, or alanine transaminase, is important in the alanine cycle by transferring an amino group from the alanine to alpha-ketoglutarate, which converts the alanine into pyruvate and the alpha-ketoglutarate into glutamate. Similarly, AST, or aspartate transaminase, transfers an amino group from aspartate to alpha-ketoglutarate, which creates oxaloacetate and glutamate. Since these enzymes are both in the liver, levels of both ALT and AST in serum will be elevated after liver injury or due to hepatitis. Vitamin B6 is required as a cofactor for glycogen phosphorylase, which is important for maintaining blood glucose by freeing glucose from glycogen. It's also required for cystathionine synthesis from homocysteine, which in turn is used to make cysteine. So without B6, you could become deficient in cysteine. And you also need it as a cofactor for delta-aminolevulinic acid synthase. Do you remember what that enzyme is used for? It's the rate-limiting step in heme synthesis, and makes delta-aminolevulinic acid from glycine and succinyl coenzyme A. As I mentioned before, B6 is also required for the synthesis of niacin from tryptophan. B6 is a cofactor for many of the reactions used to make neurotransmitters, including dopamine, norepinephrine and epinephrine, and GABA. The last role I'll cover for B6 is that it's used to convert histidine to histamine. Do you remember the two types of cells to make and use histamine? That would be mast cells and basophils, and this one's an example of a basophil. B6 deficiency can cause convulsions, hyperirritability, peripheral neuropathy, and sideroblastic anemia. So why sideroblastic anemia?
Remember, B6 is required for heme synthesis, so if you can't make heme, then the iron just accumulates in the mitochondria, which is pathologic, and it can be visualized as these blue spots in sideroblasts, or atypical nucleated erythroblasts, which are the precursors to red blood cells. So where would you find these cells? Only in the bone marrow. And why would you have peripheral neuropathy? That's because pyridoxine is needed to make the ceramide and myelin. So without it, peripheral nerves can become demyelinated. Some drugs like isoniazid, cyclosporine, and penicillamine can inhibit pyridoxine and cause some of these symptoms.
Vitamin B7, more commonly called biotin, is a water-soluble vitamin found in nuts, and is a cofactor that carries carboxylate, which is used in lipid and carbohydrate metabolism reactions. There are three major reactions that utilize biotin, and they're shown as the green circles in this diagram. The first is the conversion of pyruvate to oxaloacetate by pyruvate carboxylase. That's this arrow here. This is both an anaplerotic reaction for the TCA cycle, which means that it replenishes oxaloacetate if it gets too low, and it's also important for gluconeogenesis, since that starts with oxaloacetate. That being the case, which organs do you think have the highest expression of pyruvate carboxylase? That would be the liver and the kidneys, since those are responsible for almost all the gluconeogenesis in the body. The second reaction is the conversion of acetyl coenzyme A to malonyl coenzyme A by the enzyme acetyl coenzyme A carboxylase. which as you can see here is important for fatty acid synthesis. And the third reaction is the conversion of propionyl coenzyme A to methylmalonyl coenzyme A using the enzyme propionyl coenzyme A carboxylase. And that's necessary for the catabolism of odd-chain fatty acids and branched-chain amino acids. Notice that all three of these enzymes have the word carboxylase in them. If you see carboxylase, think biotin. Also know that reactions involving biotin will add one carbon to the substrate, such as by going from 3 to 4 carbons here, or 2 to 3 carbons here. Deficiency in biotin is pretty rare, but it can be caused by antibiotics and raw egg ingestion, because egg whites contain the protein avidin, which avidly binds to biotin, preventing it from being absorbed in the intestine. Symptoms of biotin deficiency include dermatitis, alopecia, and enteritis. Alright, so we're going to have lots of vitamins and symptoms to cover, so I'll keep the quizzes coming. A deficiency in which vitamin causes adrenal insufficiency? So that would be vitamin B5, which you need to make coenzyme A. Vitamin B9, or folic acid, is found in fruits and leafy green vegetables and performs one-carbon transfer reactions in the form of tetrahydrofolate, or THF. Tetrahydrofolate is synthesized from dihydrofolate by the addition of hydrogen from NADPH, which is catalyzed by dihydrofolate reductase, or DHFR. As I talked about in the first biochem section, tetrahydrofolate is important in the synthesis of pyrimidines in DNA and RNA. It was used at the very end of our pyrimidine synthesis pathway to synthesize DTMP from DUMP. As was the case with B12, folate is also stored in the liver. This makes it more difficult for people with a normal diet to become deficient, but in fact this is the most common vitamin deficiency in the United States, since people don't typically eat enough green vegetables. Deficiency can also be caused by several drugs, such as phenytoin, sulfonamides, or methotrexate. Phenytoin prevents absorption of folate. Sulfonamides inhibit an enzyme called dihydroapatite synthetase, which synthesizes a precursor of folate, and methotrexate and trimethoprim both inhibit dihydrofolate reductase. The most important time to not be folate deficient is during pregnancy, since folate deficiency can cause neural tube defects. As I mentioned before, folate deficiency can cause macrocytic megaloblastic anemia. However, it can be distinguished from B12 deficiency because there are no neurologic symptoms. Vitamin B12, or cobalamin, is only made by bacteria. None of the plants or animals that we eat can make it on their own, but we can still get it from animal products since they get it from bacteria in their bodies. It has a very specific absorption mechanism. A protein called intrinsic factor, which is secreted by gastric parietal cells, binds to B12 in the duodenum after it's been released from our protein by pancreatic enzymes. This intrinsic factor B12 complex binds to receptors in the distal ileum, which then absorb it into the enterocyte. Here, it loses intrinsic factor and binds to transcobalamin 2, which transports B12 through the bloodstream to the liver, where it's stored. Okay, so now that we have vitamin B12 stored, what do we need it for?
B12 is a cofactor for the enzyme methionine synthase, also known as homocysteine methyltransferase. This enzyme takes the methyl group from tetrahydrofolate and adds it to homocysteine, which together create methionine. Now methionine is important because of course you need it to make proteins, and also it can be converted into SAM, which is used to donate that methyl group to other molecules. B12 is also used to convert methylmalonyl coenzyme A into succinyl coenzyme A. So when do we do this? Well, this is how odd-chain fatty acids and branched-chain amino acids enter the TCA cycle, and it's also an important step in heme synthesis. Okay, so what are some ways that people can become deficient in B12?
Deficiency can be caused by lack of intrinsic factor due to gastric bypass surgery or destruction of gastric parietal cells, which causes pernicious anemia, and this will prevent the absorption of B12. Also, malabsorption can be caused by celiac sprue, which causes inflammation that prevents the enterocytes from absorbing B12. Another way is that the parasite Diphyllobothrium latum, which we'll talk about in the microbiology chapter, is a fish tapeworm that competes for B12 and lives in the small intestine. So patients with this parasite can become B12 deficient. And lastly, an absent or dysfunctional terminal ileum due to something like Crohn's disease or surgical removal will also cause B12 deficiency. Severe B12 deficiency can also cause folate deficiency, since methyl tetrahydrofolate can't be converted to tetrahydrofolate without B12. This folate deficiency can then prevent de novo pyrimidine synthesis, which is described at the beginning of this chapter. Prolonged B12 deficiency has neurological symptoms that include subacute combined degeneration of the posterior and lateral spinal columns due to a defect in myelin production. This manifests as ataxia and difficulty with proprioception and vibration sense. B12 and folate deficiency can both cause macrocytic megaloblastic anemia, which results in red blood cell mean cell volume greater than 110, and hypersegmented neutrophils, like the ones you can see in this image. Okay, so if we have a patient who is B12 deficient, how can we tell what's causing it? The Schilling test can be used to determine whether a patient's B12 deficiency is due to malabsorption, pernicious anemia, or a dietary deficiency. In the first part of this test, patients are given radio-labeled B12 orally, which I'm showing as this green-bordered B12 here, and also an intramuscular injection of unlabeled B12, which is this one here. The unlabeled intramuscular B12 saturates the transcobalamin receptors in the liver so that any labeled B12 that is absorbed will be excreted in the urine. A normal test is greater than 5% excretion in the urine within 24 hours, and an abnormal test is less than 5% excretion. So what could cause a test to be abnormal? Well, a normal test could indicate that the patient's B12 deficiency is due to insufficient B12 in their diet, since they're able to absorb it normally, whereas an abnormal test indicates a problem with absorption. So if you're not excreting it in your urine, that means that it went straight out the digestive tract in your feces, since you never absorbed it in the first place. So if less than 5% of labeled B12 is found in the urine, we know the patient wasn't absorbing it. So how do we now figure out why the patient isn't absorbing B12? Well, to determine whether this is due to a problem in the intestine or lack of intrinsic factor from the gastric parietal cells, the test is repeated, but this time intrinsic factor is given orally with the B12. If this now produces a normal test, it means the patient has pernicious anemia, whereas if it's still abnormal, there's probably an absorption problem in the intestine.